Hey folks, Richard here from Escarpment Labs. If you brew IPAs, you know hops are expensive, and getting the most out of them is more important than ever. What if your yeast could help boost hop flavor, extend shelf life, and even unlock totally new flavors and aromas? In this video, I'm going to walk you through the science and real world examples of how modern yeast development is transforming IPA brewing from hybrids to lab evolved to engineered strains. So let's get into it. Your hops deserve better yeast, transforming IPAs with science. So, why do we need new beer yeasts anyway? Why can't we just stick with the old classic California ale? Well, hop forward beers are really popular. For those of us who brew hazy IPAs, we know this, right? Customers crave these flavor profiles. IPA is one of the most dominant, or is the most dominant beer style out there. Remains so, at least in the craft beer space. And it doesn't really seem to be going anywhere. There are other things like lager that are certainly gaining some momentum, but West Coast IPA, hazy IPA, IPA, it's not going anywhere. These are remaining really popular, so they're a really important part of the production portfolio for any brewery. And the amount of hops we use in the beer is on the rise too. I will admit that data is a little old at this point, but I do not think that the hop utilization in craft beer has gone down. If anything, brewers are finding ways to cram even more hops into their hop forward intense IPAs. So that means that there is some cost pressure for this beer style. And that's a challenge because hops are expensive, especially when you look at some of the really intensely flavorful proprietary hop varieties. They can be really expensive and make it really quite a challenge for the brewery to make money on these beers. One of the benefits of IPAs is you can turn it around a lot faster than a lager. One of the big challenges is that hops are expensive. So, what if we had a solution to that problem? Well, it may be possible that flavoring by yeast and flavor enhancement could help you get more value out of your expensive hops. And the cool thing about yeast, I'm a yeast person, I'm biased, but the cool thing about yeast is that yeast is not as expensive and you can reuse it. You can't reuse hops, you can't reuse malt. So yeast has this crazy advantage. It's horrible for businesses like ours, but for you as the brewer, you could actually reuse the yeast if you treat it right. That's crazy. So, why aren't we using new yeasts in all of our beers? Sorry, I just had to move my face a little bit. Um, we have this kind of funny scenario where the most popular beer yeasts out there have not evolved or improved in decades. If we look at the California ale strain, for example, that was really kind of set in stone in the 1980s. What is now one of the most popular ale strains out there, your Foggy London Ale, London Fog family. That yeast very likely hasn't changed that much since the 1970s. Just so happened to be that this yeast is really effective at holding a stable haze in a highly dry hopped beer. They weren't making those beers in the 70s, but it just so happens that this random yeast does a really good job of that. So now it's one of the most popular ale strains worldwide. And then, of course, your 3470, your classic lager strain, that has really dominated the beer industry, especially on the lager side. It's probably the most popular yeast strain in use for beer globally. That has not changed that much, certainly not in the last 50 years. So we're kind of running on historical strains, which is, to me, it's a little bit of a strange situation because, if anything, it's actually easier to develop new yeast strains as opposed to our other beer ingredients like malt and hops. Those things take years or even decades to develop new varietals, whereas a yeast, I'll tell you right now, you can make a new yeast strain in a weekend and have something different. So we're in this funny scenario where even though yeast science makes it easy to make new strains really quickly, we're not really there in terms of the commercialization, or at least we weren't when we were looking at the beer industry 4 or 5 years ago. So the Escarpment Labs approach was to just look at what was out there, do a survey, look at the data. So this is data from, it feels like ages ago now, but this was data from 2019. We took a bunch of different brewing strains. This did include some of the farmhouse beer yeast like Kvike that we had access to at the time. And what we did is we put them all through an identical fermentation process. We measured aroma compounds using a gas chromatograph, and we put them all on this plot, which I like to think of as a flavor map. Each position on the map corresponds to a number of different flavor compounds. So you can look at this map as two strains that are close together have similar flavor profiles. If they're further apart, they have different flavor profiles. Um, to the right here, you have strains that are more fruity. This positioning on the map is associated with fruity flavors, those esters produced by the yeast. Um, Going south on the map, you have the spicy or Belgian flavors. So you can see all the strains on the south part of the map have that POF, phenolic off flavor, spicy clove aroma, which might not be desirable in an IPA, for example. Everything to the left is your more neutral flavors because, again, it's not corresponding to fruity esters. And then I think of importance to IPAs, strains that are sort of up here in this upper right quadrant of the map are your strains that are going to be associated with high degrees of biotransformation. So at the time we were measuring terpene biotransformation, and we will get into why that's important. But interestingly, not a whole lot in some of the desirable regions for yeast development, especially if we were looking at the way that the IPA was developing. At the time, the hazy IPA was really, really starting to become dominant and take off. And yet there weren't a whole lot of yeast strains that offered that combination of fruity esters and biotransformation. You have some strains offering some mild biotransformation and aromas. You can even see, Foggy London, the old standard over here, not really that fruity, does have some biotransformation traits. And then some other strains that you wouldn't expect, like Ringwood or, Hefeweizen strains, showing certainly some promise there. But really, this was surprising to us. There's no real strains that are naturally ideal for some of these modern IPA targets like high biotransformation and fruity esters. Of course, haze became important as well. So that became an important trait to try to develop yeast for. So opportunity, uncharted territory, right? That became a big focal area for us with our own yeast development. So how can we make new IPA yeasts or improve the existing ones so that brewers like you can continue to innovate? We have a few different options. What's really cool is that we have a number of different established methods to develop new yeast strains for beer. So that can include classical methods like breeding. It works a lot like plant breeding, for example, or even just making types of dog breeds. You can take a Labrador and a poodle, make a Labradoodle. Um, Lab evolution, which is basically doing what a brewery does, repitch the yeast, but we can do it on an accelerated timescale, and gene editing as well. So I'm actually not going to get into gene editing in this video. We will have a separate video on that topic because it is quite cool. It's something that we've been getting into a lot more recently compared to these other methods. Okay, so let's talk about hybridization. This is one of my most favorite to talk about. I think it's also a lot of potential to use hybridization to develop new beer strains for targets like IPA or even perhaps for lager. Hybridization is essentially combining two different strains of yeast through laboratory methods to develop a new single strain hybrid yeast that has genetic information, DNA, from both parents. So I could get into the nitty-gritty details. I will a little bit, but just to start, we have two different strains. We can help them along to combine and to form one new strain. So that's a key point with hybrids. It's a single strain. It is not a blend. And if the lab has done their job right, it's stable as well. So it's not going to just constantly change. What's cool about the hybridization process is that yeast works a lot like mammals, for example. We have male and female chromosomes in most mammals, and different sex cells. Yeast is a little bit simpler and actually a little bit more confusing. They have A and alpha. You can sort of think of that as like the two different sex chromosomes. You need the opposites in order to form a new hybrid yeast. So if an A cell and an alpha cell get together, dim the lights down low, play some slow jams, you might just have yourself a brand new hybrid yeast strain. In the lab, it actually is a slightly more complicated process, but that's the basics of it. If you're wondering about this cartoon that I'm showing too, this is what this process of the two cells interacting with each other through pheromones and then basically extending their cells to each other and fusing is called. It's called schmooing. Yeast scientists are really creative, and so they named this process when they looked at it under the microscope. They're like, oh, it really looks like that cartoon called the schmoo. So that's what they called it. So you can go and look at scientific literature and see references to schmooze all over the place. So the yeast are constantly schmooing. How does it work in practice? There's two different ways that we can develop new yeast hybrids. I sort of think of it as like the really precise way and then the more like quick and dirty way. So precision mating, otherwise known as spore-to-spore yeast mating, does require more equipment and more materials, but it is a more precise and fast method. So you will typically see this method in more sophisticated labs. You have to have like a fancy microscope with the tiny, tiny needle that can pick up individual yeast spores and stick them together on a grid. But it's really, really efficient. So what you do here is you take your parental strains that you want to combine. Usually you have one strain that you want a certain trait of and another strain that you want another trait of, right? Like, you might want uh, the voice of Celine Dion and you want the height of Michael Jordan. Okay, let's put them together. Let's make something new. We can do the same thing with the yeast. We might have one that we really love the flavor, another one where we really love the stress resistance and the flocculation. Okay, let's put them together, make a new hybrid, make something better than the parents. So we take those strains, we put them through the sporulation process, which usually involves starving them. So they kind of freak out, they go into survival mode. These spores are actually a survival mechanism. The yeast goes into sporulation basically as a last ditch effort to survive. And the cool thing then is these spores can then recombine with other spores and actually introduce new diversity into that population. That's how this works in the wild. What we can do is we can manipulate this process. We can make the yeast make spores. We can break down the walls of those spores. We can use a fancy, tiny, tiny glass needle, pick up those spores, stick them together on a little Petri plate, and let them grow. And all of a sudden, we have brand new hybrids that we can then screen to see if we've made something that meets our desired targets. So cool method. The other one that it works just as well is called rare mating. And what's cool about this one is you can do it with relatively limited equipment. You do need some fancy chemicals, but it's really just you need to make agar plates, Petri plates, and have some basic microbiology techniques. So here, what you do is you take the parents and you make them worse. So you expose them to chemicals that make them unable to make certain amino acids. So this strain can no longer make uracil. This strain can no longer make lysine. We stress them out. We mix them. Um, they form what we literally rare mating. Some of the cells will mate. It's just a numbers game. We're putting millions of cells into a little test tube and letting them do their thing. And then what we can do is we can actually screen for any strains that have figured out how to uh, make their own uracil and their own lysine, for example. So that would only be the case with a hybrid. So we have a nice screening method. It's all based on agar plates, and we can select for hybrids this way. So that's a great method to make hybrids in a little bit more of a low-tech way. So that was probably my most complicated section, just so you know. If you're like, brain is melting from the science, like we're pretty much done with a lot of that. So just to give an example of a strain that we produce through breeding, the Lalbrew Pomona. So Pomona is a strain that Escarpment Labs developed. It's now available globally through Lalaman Brewing. That is a product of hybridization. So in this case, we used yeast rare mating to combine two strains that we really liked. One is a British strain, a UK genetic strain that has really efficient fermentation and fairly neutral flavor. So we really, really like that strain, just as a basic fundamental ale strain that does a lot of things. It works great to breed with other yeasts as well. So we've worked with that a lot. And really what we're trying to do is get the flavor of the other parent. So that other parent is in the beer 2 family. If you've seen some of my other videos, you'll know what that means. That's the more flavor positive yeasts. That includes a lot of the Saison and Belgian yeast. They tend to have much higher production of esters. They also tend to have quite strong hop biotransformation. So this parent did have some limitations. Luckily, it was not phenolic, so we already got that out of the way. But it is STA1 positive. It is diastatic. So most brewers in the modern age have moved completely away from STA1 positive strains. So um, breeding that out was a big target for us with this project. But that strain does have really excellent aroma formation, those tropical fruity esters and that biotransformation. So we were trying to get something that combines both of the parents. One of the other cool things about hybridization is that you have this phenomenon called hybrid vigor, where it's not even that you get something in between the two parents. You can have hybrids that will outperform the parents. So we see this in crop breeding all the time. You can see, for example, this hybrid corn has well, it's bigger than the two parental corns, and we see this in yeast as well. So just to give an example from a breeding project that we did, you have two parental strains here in red and blue. It's a fermentation curve in this case, and the hybrid strain is outperforming both of them in terms of attenuation and in terms of fermentation rate. It's a subtle difference, but we're still seeing that hybrid vigor, and we've seen that multiple times across multiple projects where we go and we breed brewing strains. So it's a really important effect where we can actually get something greater than the sum of its parts sometimes. So thiol libre is a great example as well, where that strain was bred for thiol release, and it has more thiol release than either of its two parents. So I'm burying the lead a little bit here. We have a few different examples of hybrid yeasts that we've developed at Escarpment Labs. Jotun was number one. We crossed a Hornindal Kvike with a French Saison to make a Saison yeast that's more flocculent. I would say that's a fairly niche application, but it's a really cool strain. Hydra has found a place in IPAs as a low attenuation strain. It's great for breweries that have single infusion brew houses that are struggling with hop creep and trying to get that if you're aiming for a more juicy, sweet IPA, this strain is a great option. And it's a hybrid of Vermont and Cerberus or Sactois. So two strains that are already quite nice for IPA production. And then Thiol Libre as well. We've talked about that one in the past, but that's a hybrid of Kvike yeast with a wild yeast isolated from guava. And just so happens, because life imitates art, I guess, it produces these guava, thiol tropical aromas, and that's another hybrid yeast. Even more that have come out in recent years. Elysium is a hybrid of Kvike, once again crossing in Cerberus. You can see that we have some favorite strains for breeding. That's a really cool strain if you want to pump up the pineapple ester, ethyl hexanoate. Really does a great job of aroma production. I've had Elysium beers at 5-6 months that still have like really prominent pineapple. It's honestly a fantastic strain for IPAs, sours, a lot of other beers. It's kind of under the radar right now. And then I would say a much bigger hit, the Loud Brew Pomona. That's another product of breeding, as well as Lab Evolution, which we'll get into shortly as well. But that is a hybrid of a clean, efficient yeast with a really flavorful yeast to produce something that's more compatible with modern IPAs. And that brings us to adaptive lab evolution and my favorite scene from Jurassic Park. Life finds a way. That's it with evolution. You see it in Jurassic Park. You see it here with yeast. Life is always evolving. It's always challenging its environment. It's always being challenged by its environment, and we can actually tap into that constant evolution in the lab and in the brewery to improve our yeasts. And I think that's really, really cool. Shout out to Jurassic Park. Okay, so here's an overview of how it works in the lab. This looks maybe a little complicated, but at its most basic, it's very, very similar to a brewery just constantly repitching their yeast and picking yeast from the best batches of beer. So if we want to evolve a strain, first of all, we have to choose some kind of environment to challenge it. So that might just be wort, that might just be beer, right? If we just want to make that yeast more efficient in beer fermentation. Or we could choose high sugar. We could choose toxic compounds like terpenes or alcohol or hops. One way or another, we can create a selective environment to try to force that yeast to evolve over time. Because random mutation is always happening. Every time that cell divides, there's a chance of a random mutation occurring when it makes a new cell. So we force multiple divisions, hundreds of millions, billions, trillions of new cells, we're going to eventually select for mutations that give that yeast strain some kind of competitive advantage in that environment. So over time, you will have yeasts that mutate and take over. And that's what's going on. Sometimes this happens really quickly. A lot of the times it takes a number of generations. Most lab evolution experiments for beer yeasts that we've done will be at least 30 generations, sometimes more than 100. But from a brewer's perspective, you can just look at this as sequential repitching over and over and over again. Eventually, you want to cut it off and see if you've achieved what you're set out to achieve. So in our case, we'll check the colonies to see if we're looking for yeast that grows on malt sugars. We might use an agar that selects for how fast the colony grows on malt sugars. What a brewer might do in history, this is basically how every yeast in every traditional brewery evolved before microbiologists. They would just say, hey, I really liked that batch. Let's use the yeast from that batch and keep it going that way. So this was already happening. This has been happening in breweries for hundreds of years already. We're just able to tap into the same thing in a more controlled manner in the lab. And hopefully at the end, you end up with an improved strain that has some kind of beneficial trait. It's actually a really simple method. Really, all you would need is you can do it, at home, even as a home brewer. If you want to just make a starter every day for 90 days, you absolutely could lab evolve your own yeast. Or you could, if you're in the brewery, just repitch a ton of times. I wouldn't necessarily advise repitching 100 times in every brewery because you certainly will see some drift. You're literally going to see the yeast evolve genetically and change how it responds to its environment. But you can do this. You can evolve a yeast. It's not some, highfalutin technique. Just to give an example of a brewery that has evolved a yeast, Badlands, a local brewery here to us here in Ontario. Maybe not the greatest customer because they bought a pitch of Foggy London Ale like 6 years ago, and then they've been repitching it ever since. So they've actually gone above 200 generations on their own house evolved Foggy London Ale, and we've been actually over time taking samples of that yeast every so often. I think we have Gen 60 and Gen 120 and now Gen 200. And taking a look at that yeast and doing a little bit of a study on how it evolves. So you can see an example here where we've looked at aroma compounds. We've measured the aroma compounds on the GC, and you can actually see that there's some differences between that evolved Foggy London from Badlands and our original popular strain. So you can actually see that it has a higher production of certain esters, like isobutyl acetate, kind of bubblegum ester, as well as a higher production of acetaldehydes. So that may not be necessarily desirable to everybody. It may mean that strain just needs a little bit longer to clean up, but certainly some differences. We also saw some differences in flocculation as well. So even though it makes great hazy IPA, it actually flocks better. So really, really cool example of a brewery deliberately evolving yeast to suit their needs better. We do it as well. So the other side of the picture for developing Pomona, Lalbrew Pomona, which is a great yeast for IPAs, was lab evolution. So we got some hybrids that we thought were okay from that project, but we thought, okay, to make this viable as a real contender for a brewer to switch to, we need to make it better with hazy IPA production in terms of haze stability. We need to make it more efficient when it faces a high rate of hops like we see in modern IPAs. And we also need to make sure that it delivers on the biotransformation. So we thought, well, why don't we just put it in hazy IPA wort for a number of generations? Just do the same thing that Badlands did. So that's what we did. In the case of Pomona, we went 48 transfers, so it's 300 and something actual generations of yeast. And then we screened what came out of that for brewing specs. So looking for something with good haze stability, good fermentation efficiency, and good biotransformation. Through all of that screening, I'm happy to report that we were able to land on Pomona, which you can use in your brewery no matter where you are in the world. So just some more of the specs on Pomona. We achieved pretty strong tropical fruit aroma. It also produces pretty distinct peach and stone fruit aroma, especially depending on the hops and grains that it's paired with. And we'll talk a little bit about why. How do you get peach very, very soon. And it's also haze positive. So that's actually really important because there's not very many haze positive yeast strains available that are also compatible with IPA production. So that's why the vast majority of brewers just stick with a Foggy London type strain. So it was really exciting to have a new strain with this trait in Pomona because I think it opens up some flavor diversity opportunities for brewers. So in addition to just efficiency, you can also evolve yeast for flavor. So one example where it's really useful is in improving terpene biotransformation. So terpenes are really important. They're kind of one of the most fundamental flavors of hops and therefore of beer in general. However, one of the challenges is we don't understand a whole lot about how terpenes are transformed in the yeast cell. It's a product of likely a few different genes or genetic pathways. But one of the advantages with terpene transformation is that you can get more aroma from the hops. So you really can get more value from the hops. You can turn floral hops into more citrusy. The really classic example I like is Centennial is a really floral hop on its own, but you pair it with the right yeast and it goes super citrusy. And Centennial is not an expensive hop to get. The transformations can give you more aroma, so that gives you a little bit more shelf life or intensity, or just be able to get a different flavor profile from your hops. If you're over contracted on something, it's great to use a yeast that can modify those flavors and get a little bit more diversity in your beers that you're offering to your customers. So Lab Evolution is a great tool to improve this because we don't really fully understand how these reactions happen. We just know that we like this flavor. We like this citrusy flavor. Just to show an example, yeast will actually convert that floral flavor geraniol into the citrusy flavor and other ones too. It's complex. You might get Nearol, which is like a lemon lime, or Linalool, which is like, I mean, I always say Linalool smells like Fruit Loops. Really, it's Fruit Loops smell like Linalool. More complexity through biotransformation is really the summary here. And another thing that's really important with terpenes is that their flavors are synergistic. So if you just have one terpene, you might have a certain aroma profile and intensity. But when you layer terpenes on top of each other, you get more intensity and you get more complexity. So that's where the biotransformation of terpenes is really valuable. If a yeast can take geraniol and turn it into three different other things, then you're going to get a more complex flavor profile. That's one of the big benefits of biotransformation by yeast. So just to give some examples of strains that have been evolved by us, we'll get back to terpenes in a second as well and building better IPAs. Terps is a strain that was specifically lab evolved for enhanced terpene biotransformation. So that's the example here for the laboratory evolution is enhancing that flavor. It does a really good job of that beta citronellol production. But let's not forget about Crispy. Crispy is a yeast that was actually just lab evolved to be more consistent. So it's a Kvike yeast, and we tried to just adapt it to some of the more modern brewing practices. And then, as we talked about, Pomona being lab evolved for hazy IPAs. So, what can we achieve with terpenes? This is just an example of where the lab evolution is really helpful. This is just an experiment where we tested a few different yeast strains, a few different suppliers, dry and liquid, for terpene biotransformation, just to get the lay of the land and compare some of this, conventional IPA strains out there with some of the strains that we've developed for IPA production. So you could see some of those differences. Luckily for us, terps are really punching above its weight, retaining some of the existing terpenes from the wort, but also producing beta citronellol, that biotransformation product. And Pomona is certainly punching above its weight as well compared to a lot of conventional strains. Some of the conventional strains like Cerberus and Vermont do quite well also. But this is really a good example of where the lab evolution is helpful because this trait is somewhat harder to engineer. You can get yeast to make terpene pathway from basil or strawberries, but then it usually ends up tasting quite artificial. So getting an integrated terpene transformation can really help to just lift and layer those hop aromas better. One more thing I'm going to talk about in terms of flavor is lactones, because we think these could be a new frontier in our understanding of IPA biotransformation, and yet another flavor that we can layer in to build more complex and interesting IPAs. So lactones, it's a whole other class of compounds that we often associate with certain fruit, especially stone fruit and peach being the classic example, as well as coconut. So these are naturally found in these fruits, especially in coconut. A lot of these lactone aromas compounds are associated with coconut, especially on their own. and they can be found in our beer ingredients. So grain does have lactones or lactone precursors, which are typically fatty acids. It seems like oats are actually quite high in some of the fatty acids that could be converted into these lactones. And some of the modern hops that have really gained some interesting notoriety for peach, stone fruit, or tropical coconut aromas, like Sabro and HBC 1019, are also appear to be quite rich in lactone. So it is a really relevant class of aroma compounds in beer, especially with modern beer, with some of these hop varietals that have had wild American genetics bred into them to get that lactone aroma, or using alternative grains in IPAs. We were able to show that in some of the work where we were doing some field trialing of Pomona. So both Pomona and the Lalman Verdant strain, we were able to measure noticeable levels of lactones in a dry hopped IPA. So they're there and they're relevant. And why lactones might be helpful, especially if you're trying to chase that peach aroma, which I know for a lot of brewers has been, really fleeting. It's really hard to consistently get peach in an IPA. One tool that you can use, one framework to help improve that, is to understand that this is a product of synergy. As we talked about, lactones on their own can be kind of coconutty, but if you combine them with other aroma compounds, you can increase the intensity of that flavor perception. So this is some data from a really great paper, showing how to impact the fruit aroma perception of beer. And one of the conclusions from this data is that if you combine multiple classes of aroma compounds, including lactones, esters, and terpenes, you can increase the perceived intensity of some of those really challenging to produce aroma profiles like stone fruit and peach. So if you're a brewer, you're looking to get that peach aromatic in your IPA or other beers, I would look to a process and a yeast that's going to deliver you all of lactones, terpenes, and esters. That's going to give you the best shot at that consistent peach aroma. And that seems to be the feedback from a lot of brewers that are using Pomona, especially in combination with certain hops. I will suggest, for example, Simcoe works great. And of course, some of those peachy naturally hops like HBC 1019, etc. So that brings us into the second section of this presentation, where we get practical. We're going to talk about how can you use some of this new science and new information, new strains, to design a better IPA. First off, how do we select ingredients for biotransformation to maximize those aromas and make the most cost-effective beer? So number one, we want to look at terpenes. I like to think of terpenes as kind of the base layer for IPA flavor. They're always there. You might have other things like thiols and esters kind of dancing on top, but terpenes are that really, really important base note. So you can look to hops that are naturally quite terpene rich, especially hops that have some of that geraniol that's going to be able to be converted by the right yeast to citronellol and other terpene compounds, because you're going to be able to build that base layer, and if you have multiple different terpene compounds in that aroma base for your IPA, it will typically mean that you have a more complex end result for your customer. The cool thing here is that you don't have to go with the hottest, newest hops here. Classic C hops like Centennial, Cascade, and Chinook, they work great. You also have hops like Strata, some somewhat newer hops that can work really, really well here too. Basically, any hop that's described naturally as dank is a really good candidate to be biotransformed into more citrus. And there's yeasts that can do a great job of that. Pomona, Terps, as well as some of the classic strains like Cerberus and Vermont. When we're looking at thiols, and I haven't talked too much about thiols, I think we're going to do a separate video on thiol biotransformation, because this is one of those things that really benefits from strain engineering. But if we want to maximize thiols, which are important, they're kind of that like top note, that treble, that lead vocal. They can add a lot of character, personality, and flash to an IPA. They add these tropical flavors. Um, you can of course use hops that are high already in free thiols, like Citra, Nelson, Mosaic. Free thiols aren't free. Those are some of the most expensive hops you're going to be able to source. And usually, cost of aroma hops does tend to correspond with the amount of free thiols, because, we open the bag, they smell great. Okay, I'm going to spend more money on that, right? There is an opportunity to use hops with bound thiols with the right yeast. So hops like Cascade, Calypso, Motueka can work really, really well if you pair them with the right yeast. So you want a yeast with an active beta lyase enzyme. So examples from our catalog are Thial Libre, as well as the Super series. So Super Fog, Super Lager, those are new strains that are improved for thiol release in classic strain backgrounds. But you can also consider other sources of bound thiols. So your malt matters, adjuncts can be used like Phantasm. You have some opportunities to boost the thiols without necessarily just putting in the most expensive hops at 4 pounds per barrel. So nothing against that, but the purpose of this presentation was to give you some ideas to reduce your cost. Timing is important with the ingredients as well. So if we're trying to maximize terpenes, typically the best approach there is to introduce the hops in the late kettle or the whirlpool. A lot of brewers are now doing what's called a cool pool or a cooler whirlpool. So typically I see that around 70 to 80 Celsius. So a lot cooler than an ordinary whirlpool would be closer to 90. That can really help you to maximize your terpene extraction without getting as much isomerization of alpha acids and bitterness. Um, Dry hopping early can also expose the beer to more terpenes and get more of those into solution as well. So they're not the most soluble compounds. So earlier and hotter is always going to be better to get those soluble and able to be transformed by the yeast. Thiols, it's you kind of have two options. You go right at the start or right at the end. If you have bound thiols, you can actually do a mash hopping process. The mash hopping, put simply, can help you get more transformable thiols from your hops, especially if you're using a yeast that just has the natural yeast beta lyase in it, which in the case of all of our strains, they do. It's nothing has bacterial genes in it. You can also get bound thiols into solution in your wort through late kettle whirlpool hopping, giving that yeast lots of time to convert those thiol precursors into aroma active thiols. Again, we'll have probably a full video update on thiols as well. If you want unbound thiols, so the ones that you're already getting out of the hop bag, then dry hopping mid or end of ferment is the way to get those into the beer. Haze, this is another key factor. You either want haze or you don't. You never really want the opposite. You either want haze in your hazy IPA or you want your West Coast IPA to be clear. So you have some options here. If you want to add haze, you have options for yeasts that are actually haze positive, and this can make a big difference in the stability of the haze in the beer. There are other factors that matter as well. So that haze, it's proteins, it's polyphenols. So if this yeast is stressed out, it's just going to break down those proteins. So you need the yeast to be healthy. Late dry hopping is correlated with stable haze as well. That's been shown time and time again. Dry hop content, so loading up more of those polyphenols and those hop oils can also help to get a more stable haze. And a haze positive yeast. So haze positive yeasts tend to secrete a higher amount of certain manoproteins, so proteins that can bind to other proteins and polyphenols and form a stable haze. So Foggy London and that London Fog family, those tend to be the classic options. You have Verdant as a dry strain, Pomona. We've developed other ones as well. So, for example, Hydra is also a haze positive strain. If you want a clear West Coast IPA, but you want to use modern dry hopping rates, that's going to be a challenge. So you do have some tools at your disposal. You can do early dry hopping that has actually been shown to reduce haze. If you dry hop, at knockout, day 1, day 2, early, that can really help. Of course, less hops will get you less haze. So if you're doing a West Coast IPA, don't hop it like your hazy IPA. Move more hops into the whirlpool. You need lots of hops in the whirlpool to get that nice complexity of bitterness and also to get lots of soluble terpenes. And also that'll help you with your haze. And using a haze neutral yeast. So your classic California ale will work great. Terps, Vermont, lager yeast, you have lots of options for low haze strains. Super lager is awesome in a West Coast IPA. I know a brewery that's using super lager in their West Coast IPA. It works awesome. Other options to improve your IPAs, just based on a lot of conversations and a lot of listening to what people that make really tasty IPAs are doing. Cool pooling, as I talked about, that colder whirlpool step can really help you to get maximize your terpene extraction. Works really, really well, especially for West Coast IPA. But now I've seen that practice moving into hazy IPAs and other styles as well. Another one to look at is I love this term, cone shaving. A lot of the more old school brewers do this. It's a practice that I think has kind of been lost over time, but it's really useful. What this is dropping the trub from the fermenter, as well as some of the early flocculating yeast early on in the fermentation. So 24 hours, 48 hours, for example. This is actually really genius because trub is great for yeast, but it's really bad for flavor stability of beer, especially hoppy beer. So what this does is you give the yeast access to the trub while it's growing, while it's going through lag phase and log phase. And then you just take the trub out when the yeast no longer needs it. So it has been suggested that that can help reduce mercaptan or just unpleasant sulfur aromas. It also means that you're removing a lot of the metals that are bound up in the trub, like manganese, that can be really, really bad for aroma stability in IPAs. So this is if you're not doing this, I would give it a shot. You might be able to get more hop aroma just through dropping trub once the yeast has absorbed it. Also put this together, and we'll put a copy on our website as well. But there are some tried and true options for yeast and hop flavor combos. I don't want to say that this is the only option, but I just want to give some ideas for how you can combine some of these new yeast strains with hops to get more targeted flavors. I'm all for complexity, and you'll achieve complexity as well, but it's also super useful to say, I'm going to make an IPA with pineapple aroma and actually have it come out with pineapple aroma on the first shot. So, just some quick suggestions. Hydra is your go to option for mango. Uh, this strain produces a lot of esters and terpenes that tend to associate with the flavor profile of mango, especially if you combine it with hops like Strata. I like Vista as well, and that's a public variety. Certainly Citra too. Elysium is our pineapple pusher, especially in combination with hops that are fairly terpene rich, because you're going to get a lot of this pineapple ester from the Elysium. Some of these terpenes coming in off these other hops, or even just actual, pineapple aromas like Sultana, can really help to support that. And you don't have to use, crazy fancy hops with Elysium. Thiol Libre, still quite a great option for guava and passion fruit. It does that thiol ester profile really, really well. And the cool thing is that you don't have to use super fancy hops. You can use Cascade or Calypso, for example. Pomona, that's our peach. It does a great job at producing peach aroma, among other things. Like I've seen so much diversity and potential from Pomona, just depending on how brewers use it. But certainly, one of the first things that I would try with Pomona is pair it with something like Simcoe or Nectaron or HPC 1019, because you can get a really nice peachy aroma profile from it that I think is just really tasty. Terps certainly pushes the citrus. Get a really nice tangerine note from that strain. I like it with some of these really sort of terpene-rich hops. So, Crush, Idaho 7, or even just classic C hops. You have a lot of option to make some really, really nice beers using terps. And then for a newer strain, Super Fog, it's a Foggy London that has a more active thiol biotransformation. So being able to get some of those more tropical notes, but layering that with more typical hazy IPA hops. So if you're making a Citra Mosaic or Citra Mosaic Nelson or Mosaic Nelson Citra IPA. That yeast can certainly help to layer some more aroma in there. Another cool thing about Super Fog is that it has slightly enhanced glycerol production. So really great option to kind of combat the beer drying out a little bit from hop creep. Getting some glycerol in there and building back in mouthfeel and body. So that's a really nice strain to try out for hazy IPAs. Going to close this out with some recipe ideas. So let's put it all together into some ideas that you can use. And hopefully my goal here is that you walk away with some ideas for how to make your next beer. So just to show some examples of how I would think through this, let's make a recipe for terpene biotransformation. So we can pick some yeasts. We can pick some hops that we know are going to be terpene rich up front. So looking at something like Strata and Simcoe, I think that's a nice combo. You get a little bit of thiol in from the Simcoe. Strata's got a really, really nice terpene profile a lot of the time. We can put those in the whirlpool, really load up a lot of geraniol, and that can be transformed by the yeast, as well as 4-MMP, that sort of grapey thiol coming in off the Simcoe. I find that tends to be a lot more stable in beer, so it works pretty well on the hot side. Fermentation. I think Pomona is your perfect pairing here. Going to get a lot of peach. Going to get a lot of stone fruit and certainly some citrus from the terpene biotransformation. We'll have a really good base that we can kind of punch up with dry hopping. So this is a case where I'd probably still look at some fairly typical hops like Citra and Simcoe. Use those towards the end of the fermentation for haze stability. Keep the time short. Try to preserve those aromas, especially with Citra, where you're getting so much volatile thiols coming in off of it already. And I think you could probably use a lower hopping rate than you might normally use for a conventional haze strain. Here in the dry hop, you could probably reduce it, somewhere on the order of 25 to 40%. So you'd be able to achieve a pretty efficient IPA recipe and process with this kind of combination. If we want to look at thiols, and as I said, we'll do another video on thiols just to give a bit of an update. Here's where we can have some fun with selecting hops that are high in the thiol precursors. So something like Cascade or Calypso. I would even just try just straight Cascade. Depending on the direction we're going in our beer, I do tend to prefer the thiol forward beers like showing that aroma, although you can certainly layer heavy dry hopping with a yeast like Super Fog. But if you want to show something that really shows that thiol aroma, and I think it actually can work quite well to make lighter hoppy beers like pale ales, for example. Using a yeast like Super Lager, Super Fog, I would certainly look at Super Lager in a West Coast IPA. I think that's where it shines as a thiol enhanced lager strain that's not like artificially intense. Super Fog, great for hazy IPAs. Thiol Libre, kind of general purpose. Like really like it in a lot of different applications where the thiols are useful, like blonde ales, sours, and IPAs as well. Got lots of options there. You can certainly punch up thiols even more with aromatic hops. But yeah, keep in mind that you're always kind of trading off between the yeast aroma and the hop aroma. I would look at the thiol transformation again as an opportunity to reduce the amount of dry hops that you require. You could even, I've seen successfully pairing the thiol release hop yeasts and process with hop aroma extracts that are more enriched in terpenes or even just terpene extracts themselves. There's a lot of those on the market as well. So some opportunity to have an even more efficient process that can work really well pairing terpenes with some of these thiol enhanced yeasts. So hopefully that was useful. Hopefully you walked away from here with a couple ideas and a better understanding of how we can make our IPA world better through science. If you do want to learn more, make sure you check out our knowledge base and our blog, as well as right here on our YouTube channel. I'm sure right after this video, more will pop up. Feel free to continue to educate yourself on brewing science and yeast. And really happy to have you here if you've made it all this way into the video. Let us know what you want to learn next in the comments, and thank you for listening.